Hi dear students. We came across the different types of mutations in DNA in the previous sessions. And of course we know various animate, inanimate as well as various animated movies that we come across with mutations, like we have Hulk, we have Spider-Man, and we expect X-Men all to be our heroes based upon the mutations that they have obtained. But even the angry Hulk sometimes needs to become a normal person in his life. So, the drastic effects which could be caused by some mutations might be also needed to be reverted back to a normal situation where the damage is not caused by the DNA mutation, it does not exist. So, if you go to analyze different organisms, it has been found that they themselves have various mechanisms by which the mutations that have been caused in them could be reverted. These include various natural mechanisms, and as we have previously known, we know that different types of agents, whether it be physical agent, a chemical agent, or even because if there is an error during replication, mutations can happen. So, for the well-being of an organism, the system always tries to make up some strategies so that they could overcome such mutations. And today we are going to learn about the DNA repair mechanisms which could be present in various microbes as well as in certain organisms too. So let's go into the details of that in the coming sessions. DNA repair. The first type of mutation as well as its repair, which I would like to discuss, is based upon photoreactivation. The photoreactivation based DNA repair was mainly discovered by Albert Kellner, and this is the scientist behind this. Now, what is photoreactivation? But before that, we need to talk about the UV-induced dimerization. I hope you do remember that the UV exposure results in the formation of certain dimers, like you have sometimes thymic dimers which are mainly formed as a result of UV exposure in microbes, and we do use this in the laminar air flow also to kill as well as to sometimes even to mutate the microbes. So now this is, you can have a close look on this. Sometimes if the thymines are being close to each other, a thymin-thymer dimer can be formed. And you know, when a thymin-thymer dimer forms, as a result of that, the mismatched base pairing can happen, as well as it could result in certain mutations of the organisms. Now, if you go to consider lower forms such as E. coli, they have a particular enzyme called the DNA photolyase, which is mainly produced by a gene called the PHR gene. It binds to these dimerized thymines. And when a normal light forms, a normal light shines on the cell other than the UV, when a normal light shines on this, the enzyme breaks the dimer bond with this light energy. And then the enzyme will free from the DNA. And this enzyme thus reverses the UV-induced dimerization. But we can see that such a repair mechanism is present in almost all the organisms, but photoreactivation is not present in all the organisms except for the placental mammals. Okay, in human beings we do not have it, but in the lower forms the photoreactivation based DNA repair of dimers is possible. Now let's go into an experiment which led to the discovery of this photoreactivation process. So I am using the videos or the animations which are being provided by the Cold Spring Harbor Laboratory, as we had previously explained earlier. In fact, it was Albert Kellner, as we discussed earlier, who discovered photoreactivation, Thereafter, various other scientists such as Stan Rupert as well as Solomon Gaudiel also played an important role in describing to us what are the factors which have been responsible for photoreactivation. So let's just go into the details of this. Gaudiel had heard about Albert Kelder's work that showed that a bacterial cell culture could mysteriously recover from apparent death by UV radiation. So what Kelder did was he took two conical flasks of bacteria and he exposed both of them with UV light. And one of the flasks was being placed in a dark, whereas the other one was being placed exposed to a day source for 1 day. And he found that he placed the cultures from the light and the dark regions on the culture plates to show that only the cells exposed to the light could recover and grow. So he took these two flasks, and next year when he noted the amount of growth was being found to be, of course, it was more prominent in the case of the light exposed than the dark treatment one. And when he took some of this and of the cultures and plated it on a culture plate, he could see that from the conical flasks which were being exposed to dark, there were no much colonies, but the ones which were exposed to light after the UV treatment showed the culturing property of microbes. So he concluded that probably something was responsible for a UV damage. And he described that as a cellular poison which was mainly being generated by UV irradiation. And later on, it was being found that it was he who did this experiment, and they thought that the cells recovered because the light helped to repair DNA damaged by the UV. I guess this is clear to you. So we will see into the next type of repair mechanism in the coming sessions.